These are your notes for the interaction of ATP and myosin. We're going to be looking at the microscopic things that are happening inside the muscle cell itself with the myosin heads and ATP, and then also we're going to be diving into the concept of rigor mortis, what happens when muscle cells die, and then also we're going to examine the neuromuscular junction and what is happening at a more detailed level than what we talked about before. So grab your notebooks. Get out, open it up to the input side of your notebook and write down this essential question. So today, essential question is to describe the process that occurs when rigor mortis within a muscle fiber, okay? I should say when rigor mortis within a muscle fiber happens. I don't know, bad typo, bad English, but describe the process that occurs inside the muscle fiber when rigor mortis happens, okay? Oh my. Alrighty, so ATP and myosin. We already know ATP is adenosine triphosphate, and myosin is that protein that makes up the thick filaments of the muscle fibers. And also, it's the myosin and actin repeated pattern that gives muscles their striations, the stripes that we see in the microscope of muscles. Alrighty, so myosin heads, they're activated by ATP, right? We know that. We talked about that already. What happens is the ATP is hydrolyzed and it goes through this process. We end up with ADP and a phosphate group. All right. So the activated heads attach to actin. We know that. And pull. That's called the power stroke. Remember, that was like step 3 of the muscle contraction process. When ADP, adenosine diphosphate, then is released, the ATP is released, has released the phosphate, and the ADP and energy is given off. The thin filaments then slide past the thick filaments with the power stroke itself. Then the ATP binds to myosin head and detaches it from the actin. Okay, so then we've got another ATP that comes in, binds, and causes the detachment. All of these steps repeat over and over if there's ATP available and calcium ion near the troponin-tropomyosin complex is high. That concentration is high, which we already knew that. This is kind of a review. Okay, so during the relaxation phase for muscles, because our muscles do need to relax. If they were tense and tight all the time, we would have cramps and it would be a very painful experience. So what do we see happen here? Acetylcholinesterase, that's a mouthful of a word. ACHE is the abbreviation. It breaks down the acetylcholine within the synaptic cleft, okay? So let's pull this back a little bit. For muscles to relax, this enzyme, anything that you see in biology or biochemistry that ends in an ASE, that represents an enzyme, okay? And oftentimes what enzymes' responsibilities are is to break down other proteins, okay? So what this does is it breaks down the acetylcholine, all right, within the neuromuscular junction in that synaptic cleft. Then what happens is the muscle action potential ceases or stops. The calcium ion channels close. The active transport pumps calcium back into storage in the sarcoplasmic reticulum. And the calcium binding protein, which is calciquestrin, there you go, helps hold the calcium ion in the sarcoplasmic reticulum, where the calcium ion concentration is 10,000 times higher than what would be in your normal cytosol, okay, normal cell fluids. All right. Then the tropomyosin and troponin complex recovers the binding site on the actin. Remember, that's where it started before the calcium caused it to conformationally change and open up the actin binding site. Okay, so the relaxation process, there's a lot of stuff happening here biochemically. All right, now rigor mortis. This is something that happens whenever an individual or any animal really that dies, there is muscular rigidity that begins, which means the muscles get really, really stiff. And this it begins about 3 to 4 hours after death, and rigor mortis itself lasts about 24 hours. So if there is an individual or an animal that has died, their body's going to be really, really stiff for at least a full day. Okay? So why is this? What causes rigor mortis to occur in a person who has passed away or another animal? After death, the calcium ions, they're going to leak out of the sarcoplasmic reticulum because remember there was a pump to keep them in there, to keep that concentration out. So the concentration gradient in the cytosol or in the sarcoplasm is just, it's less concentrated outside of the sarcoplasmic reticulum. So the calcium ions are going to leak out into the cytosol or into the fluid of the cell. And then what's going to happen is those calcium ions are going to bind to the myosin heads and then bind to actin, because what does a calcium ion do? It reacts with the troponin, causing it to conformationally change and move the tropomyosin out of the way so the actin can bind. And then actin can bind to myosin. And since ATP synthesis has ceased or stopped, the cross bridges cannot detach from actin. So the myosin cross bridges stay attached, and that's kind of like, it's that contraction, it's that power stroke, and it doesn't release. And then those cross bridges cannot detach from actin until the proteolytic enzymes begin to digest the decomposing cells. So what this means is the actin and myosin are going to be bound together in that power stroke in the contracted position until some other enzymes begin to start the decomposition process and start breaking down the body. Okay? Your neuromuscular junction, we've already talked about this already. This is the connection between the axon of the nerve cell, the motor neuron, and also the muscle fibers. Okay, and this is a nice little micrograph of this. This is looking at it from an electron microscope micrograph. Here, this is a blood capillary. All right, this is a motor neuron right here. This is a branch of the axon. Remember, those axons branch off into each of those muscle fibers. The synaptic end kind of looks like a hand coming down onto the muscle fiber itself, and this is the muscle fiber or muscle cell. The pericyte right here, pericyte. All right, and the neurolemocyte, which is a Schwann cell. When we get to the nervous system, we'll talk more and more about these Schwann cells and what their purpose is. Okay, so the NMJ, the myonormal neural junction, another way to say that would be the neuromuscular junction. It's just a different play on words. The end of the axon near the surface of the muscle fiber at its motor end plate remains separated by the synaptic cleft or the gap. Okay, so now here are some illustrations of that very same thing. The structures of the neuromuscular junction include the synaptic end bulbs. They're the swellings of the axon terminals. So these things here, like I said, the synaptic end bulbs, they kind of look like fingers almost, coming down into the muscle fiber. And that's just to give it some more surface area to make contact for the neuromuscular junction to occur. All right, the end bulbs of these synaptic ends contain synaptic vesicles, okay? The synaptic vesicles are these little fluid-filled sacs that are inside there that they're filled with the acetylcholine, that neurotransmitter that we've talked about. Then the motor end plate contains about 30 million acetylcholine receptors. So inside the synaptic end bulb of the neuron, we have these vesicles, and then once an action potential moves down the motor neuron, it releases the acetylcholine, and then the muscle cell itself, the motor end plate, receives that acetylcholine, and there's approximately 30 million receptors on each end plate. So that's crazy huge. That's amazing. All right, so the events that occur after a nerve signal. Once the nerve impulse at the nerve terminal causes the release of the acetylcholine from the after the acetylcholine from the synaptic vesicles. We already know this. Then the acetylcholine binds to the receptors on the muscle motor end plate, opening the gated ion channels so that the sodium ion can rush into the muscle cell. The inside of the muscle cell becomes more positive, triggering a muscle action potential that travels over the cell and down the T tubules. Remember, we talked about that. Now we're just adding a little bit more detail there. So the action potential is because of the change in charge due to the sodium ion coming in. Okay, then the T tubules cause the release of the calcium ion from the sarcoplasmic reticulum, and the muscle cells going to shorten and generate that force. Remember that power stroke we talked about with the actin and myosin heads, that when they react together. Then the enzyme acetylcholinesterase breaks down the acetylcholine attached to the receptors on the motor end plate, so the muscle action potential will stop and the muscle cell will relax. And we just talked about the relaxation phase. All right, well, that concludes your notes for this time. It was a lot of information. Guys, just do your best as you take the notes to write them in your own words, shorten up the statements, but make sure you comprehend what you have written. Next week, we're going to be taking some quizzes and we're going to be working our way through some exercises to help you comprehend what's going on a little bit better in class, or a little bit more thoroughly in class. So be expecting that. Make sure that your notebooks are up to date. Okay, you really need this information to be successful. All right, go back and answer that essential question, and I look forward to seeing you in class. Bye-bye.